Hello and welcome to the Mutations revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. A mutation is a random change in the DNA base sequence. Mutations occur continuously and provide a source of variation. As a result of the DNA base sequence changing, most mutations do not alter the protein or only alter it slightly, so that its appearance or function is not changed. A few mutations code for an altered protein with a different shape, so the protein is no longer functional. Enzymes are protein molecules. In this case, a mutation has changed DNA base code, which results in the shape of the active site for the enzyme to change. And as a result, the substrate can no longer bind to the active site, so this has made the enzyme, which is a protein molecule, non-functional. Another example can occur which affects the structure of a protein. So if a mutation changes the DNA base sequence for a gene that codes for a structural protein, it can significantly weaken the structural protein. So this is a structural protein called collagen, and the mutation changes the DNA base sequence, which weakens the collagen structural protein, as you can see. How the mutation affects a protein. When a mutation occurs, it can change the DNA base sequence by either inserting or deleting a base. And this can change the amino acid sequence of the protein, which can make it non-functional. Insertion of a base. On the left, there is the normal DNA, and on the right, there is the mutated DNA, where you can see we have labeled an inserted base TA. Now, when we insert those bases, the DNA triplet code will change slightly. So you can see the second DNA triplet code changes from GTA to AGT. And as a result of that, it will change the amino acid proline into serine. However, it moves all the other bases along, so it will also affect the next amino acid. So instead of the next amino acid being valine, it then becomes arginine because the DNA triplet code has changed from CAA to ACA. Deletion of a base. On the left, we have the normal DNA, and on the right, we have the mutated DNA, where the bases have been deleted. You can see on the left, GC, we will be deleting, so it will not be present in the DNA on the right. And that has an effect in that it will change the second DNA triplet code from GTA to TAC, which means that the amino acid will change from proline to methionine. Mutation on a non-coding section of DNA. Non-coding parts of DNA have regulatory functions. They can switch genes off or on. If a mutation occurs on a non-coding section of DNA, it can result in switching off a gene or switching it on, and as a result, it can have a large effect on the organism. Thank you for watching. See our revision notes, the link is in the description.